So I'm going to help you try to understand how MHCs, which are receptors on some cells, work with APCs, which are these cells, the antigen presenting cells, and how those work with T lymphocytes and help them get activated, then differentiated. So first, and I wrote a simulation that you can try at home to do with this, so I will let you know what step I'm on. So the first thing I did was set up my antigens. So in step 1A, you could either use the same kind of paper, just use different ink, and you want to make sure that the ink goes across the paper, or you can use different pieces or colors of paper with the same ink. So now I'm going to simulate these antigens just randomly being spread through the body by putting them in a few different places in the room that I'm in. So I have one here, another one here, and my third one here. So if I had another person to do this with me, I would have had that person move these into different places, maybe spin me around and randomly point me to one. Now I'm going to set up my T cell with its antigen receptor. So I'm going to use this container as my T cell, and I am going to randomly choose one of these 3 pathogens to work with. Now, if I was not being lazy, these cards would all have all the same words as these, but for recording purposes, color works better. So I'm going to close my eyes and just randomly pick this one, which is step 1A, Roman numeral 5. So now I'm going to tape this receptor for SARS-CoV-2 to my T cell. So as I go through the rest of the simulation, this T cell that is immunocompetent but naive, which means that it expresses receptors for the SARS-CoV-2 virus, and it's going to look for pieces of SARS-CoV-2 virus on its APC, and in particular on an MHC1 on an APC. But for now, let's just remember that this container represents my T cell that's looking for SARS-CoV-2. So I'm going to use 3 mugs to represent my antigen presenting cells. These work just fine because you can't see through them, which is exactly what a T cell experiences. Got my T cell over here hanging out in its thymus, waiting to seed its secondary lymphoid organ. It can't see what's inside of these APCs. It can only see what's on the outsides of the APCs. So if I was going to simulate being an APC out in the world of the body, just looking for antigens, I would run around the room that I'm in and gather up those pieces of paper. So I'm going to go grab 1 or 2 or 3. I'm actually going to set up 3 APCs with MHCs to show you how T cells actually find their targets. So this APC has found an antigen, so it's going to take the antigen in, but that is not the way that it actually displays the antigen. It has to take the antigen inside of itself and rip it up. Just like that. That's antigen processing. So I'm going to do the same thing with my other APC. Takes in the pathogen or antigen, breaks it down, which means rips it up. And now take my third one for SARS. The APC ingests, phagocytizes the antigen, then rips it up. That's what we call processing. And so far, we have APCs that have taken in phagocytized antigens and then processed them. So the next step is that these APCs have to show what's inside of them so that T cells can understand or see what's inside of them. So I'm now going to use pieces of tape and numbers of pieces of tape to simulate the MHCs that will show a piece of antigen on the outside of these APCs. So I'm going to use 2 different kinds of tape. You can use just one if that's all you have, and 1 or 2 pieces. Back in a minute. So my APCs, my coffee mugs, are now showing pieces of their antigens. Remember, they processed the antigens internally, broke them down into little pieces so that they're not harmful and they can show a piece on the MHCs. So for this virus, SARS-CoV-2, which is an endogenous pathogen, it replicates inside cells. I used 1 piece of tape for an MHC1 and showed a piece of it. For MRSA, caused by a bacteria, methicillin-resistant Staph aureus, I used 2 pieces of tape to represent an MHC2, which are showing a processed piece of MRSA. And then on my last one, this was my Giardia lamblia. Cute little parasite, causes some pretty icky diarrhea. So again, because this one is also exogenous, just like MRSA, they replicate outside of human body cells. I used 2 pieces of tape to represent the MHC2. So in the next step, I'm going to seed my lymphoid organ and my secondary lymphoid organ, and I'm going to get my APCs moving through that lymph node to see if my T cell can become activated. Stay tuned. So I've outlined myself a secondary lymphoid organ. I'm using a lymph node. So remember that the antigen challenge only takes place in secondary lymphoid organs, so not the thymus gland and not the bone marrow. So I'm going to take my T lymphocyte out of the thymus gland and seed it in my lymph node, which just means that it goes to the lymph node and stays there and waits, twiddles its thumbs, its figurative cellular thumbs, and waits for the right antigen to come along so it can get activated. So how does that antigen come along? The antigen will be displayed, like I showed you in the previous step, on an APC in an MHC1 or 2 receptor. So I'm going to first work with this antigen. This is my Giardia lamblia on, I got 1, 2 pieces of tape, so on an MHC2 because it's exogenous. So it's going to come in through this EFFerent vessel of the lymph node. The T cell is going to look at it, and the T cell is going to say, this one's not mine. So using colors, this antigen's yellow, I'm looking for blue. And now this APC travels out through the afferent lymphatic vessel, and then it goes and travels around the rest of the body. It might come in and out of a different lymph node. Maybe that's where it finds its match, or it circulates around and never finds a match. Now I'm going to try a different antigen. So this time I'm going to go with my MRSA antigen. Still displayed on an MHC2 receptor on this APC. Remember, the cup is my APC. Still has more processed antigen, but remember, the T cell can't see through here. It can only see this. So now my APC is going to come into the lymph node. Hello, T cell, do I have what you want? Why, no, sir, you do not. So my T cell that's looking for a blue antigen sees pinky antigen and says, no, thank you. Good day, sir. and then leaves through the efferent arteriole. Yes, it's late on a Friday night. Now, I'm going to go to my third APC showing its antigen. Now this time, we've got the SARS-CoV-2 virus, endogenous pathogen, because it has to replicate inside of this APC. Still have pieces of processed antigen in there, but again, T cell can't see those. T cell can only see this, that piece of antigen on, an MHC1 receptor, my single piece of tape for my MHC1 showing a piece of my endogenous pathogen. So my APC moves again through the efferent vessel, says, hello, T cell, and T cell says, yay, I found my match. So at this point, the APC and the T cell receptor are going to bond to each other. And then there's a second step that has to happen for T cells just to make sure. It's literally a double check for T cells to make sure. And that would be the binding of one other molecule from the T cell to the APC, which is called a co-stimulatory molecule. Co just means with. Once that happens, the T cell's activated, and then it's going to go on to do differentiation and cloning, which I'll show you in just a minute. Okay, so now we have an activated and immunocompetent CD8 cell. Now it is going to have to differentiate. And when we talk about differentiation, that means that the cell matures and becomes something even more specific. In the case of CD8 cells, you can get from that cytotoxic T cells and memory T cells. So this is the differentiation part. This one cell that's a CD8 that's not specific can now become more specific by expressing more receptors to become either a cytotoxic T cell or a memory T cell. Now the very last step in this is that these T cells can't stand alone. One T cell does not make an army. So these T cells are now going to go through cloning, which is the scientific way of saying they do cellular photocopying or have babies. So this cytotoxic T cell, when it clones, makes a bunch more cytotoxic T cells that are specific for that antigen, in this case SARS-CoV-2. The memory cells will also make clones of themselves, so memory cells again for SARS-CoV-2, and that is cloning. Again, think of it like cellular photocopying, so that this T cell, or every time you make a T cell, you don't have to go back to the very beginning of this video and go through all those processes. These memory cells are the value of secondary immunity. So if you survive SARS-CoV-2, you're going to make all these cells, and then the next time that SARS-CoV-2 comes along, boom, that memory cell says, I know who you are, I'm going to get you real fast. So that was for CD8. Now let's look at what happens for differentiation and cloning for CD4. So I'm now going to run the simulation for a CD4 cell that is looking for the notorious MRSA. So we've seeded the lymphoid organ with this immunocompetent but naive T cell, which means it knows what it's supposed to be doing, we think, but it hasn't been tested. The tested part is the antigen challenge. So I've still got my 3 APCs, one that's got SARS-CoV-2 on an MHC1, another one that has Giardia lamblia on an MHC2, and then my third one that has MRSA on an MHC2. So just like before, these antigen-presenting cells are going to move through the EFFerent vessel of the lymph node, say, hello, T cell, and T cell says, no thank you, you're a pink antigen, on you go. And then I have my Giardia lamblia presented on an MHC2. Hello, T cell. Yes, no thank you, goodbye, you're not my antigen. But then along comes MRSA, I've been waiting for you. So again, the T cells receptor for MRSA will bond to the antigen on my antigen presenting cell. They bond. I get co-stimulation, binding of another molecule between the T cell and the APC. And then this T cell is now considered activated, a CD4 activated cell. And then my APC can go away. So now my activated CD4 is also going to differentiate into its effector cells, the cells that carry out the effects. So I'm just going to back it up here so we can see. So the effector cells for your CD4 are your helper T cells, your memory T cells, and remember these are all specific for MRSA, not for anything else, and regulatory T cells. And then sometimes you should know regulatory T cells are still called suppressor T cells rather than regulatory, but they are effectively the same cell. So now that I've got these three cell lines of effector cells, they need to clone so that there's enough of them to effectively combat disease. So my memory cells, we're going to do some cellular photocopying and make a bunch of those. Same antigen that they're looking for, but not the same as the original cell. And then my regulatory T cells, which literally regulate immune responses up or down. So I've made some of those. That's my cloning. And then lastly, I'm going to clone a couple of helper T cells, right? Not the same as this original cell back here, but the babies of this original cell, which can do exactly the same kind of actions. So to review memory again, the value of a secondary immune response, right, what happens after you have a successful vaccination, is that you get a whole bunch of these T cells, that would be these that are specific for MRSA. So the next time that MRSA circulates through this body, boom, this T cell knows exactly what to do and will combat it and take it out immediately. So I am also posting some directions for how to do this simulation. If you'd like to do it yourself, you can actually do it in a yard or in a house and sort of play tag with animals or kids if you like, but you don't have to. Hopefully this helps a little bit with APCs and MHCs and the whole process of T cells becoming mature and effective fighters for human bodies.